Welcome to what I've heard is going to be the most sought after session. I take that on good authority. My name is Samir Das. I am the Associate Director of Technology at the McGill Center for Integrative Neuroscience at the Montreal Neurological Institute at McGill University. I have trouble getting that straight sometimes. I'm also very much part of INCF, and there's a Standards and Best Practices committee which is very on topic here. It's led by the very talented and knowledgeable Marianne Martone. And so I guess what I want to do before I introduce the rest of the speakers is just say a few words about where I'm coming from. So this isn't actually supposed to be happening. I'm supposed to be playing hockey and thousands of people are supposed to be watching me, but instead I'm chairing a standardization session. That being said, I've come to realize how important something like this is. It's very much part of our everyday lives in every single way, whether it's in construction or whether it's with technology. You want to attach some USB keys or whatever to your machine or plug in your Apple iPhone. There's all sorts of standardization or lack of, in some cases, there. With the data sciences as well, whether it's ontological standardization or other interoperability kind of aspects, APIs, you name it. There's all sorts of stuff here that is very important in accelerating science and technology and, all of those kind of things. So in fact, I've spent quite a few years dealing with standardization issues. You could actually say that I've gone into the weeds a fair bit. I think back to some of my early days with the pediatric normal brain development study and dealing with data dictionaries. Woo, that was much more complicated than I thought it would be. Even today, if we fast forward, I'm at the Montreal Neurological Institute and we're going open. We have our own open science initiative, and we are actually the first clinical/research institute that's putting out clinical/research data. So this is a very cutting edge area. And so right now, though, I'm, as we speak, dealing with standardization issues and making sure that that data goes open. In fact, sometimes I think of it as the bane of my existence, but we are not here to discuss my feelings and my personal, opinions on this. So what I'll do is I'll say a few introductory words about standardization, and then I'll introduce the speakers. So very quickly, standardization is something that I can tell you funding agencies are very interested in. They've lost a lot of money on data that's not interoperable over the years. So many of you who are scientists or researchers are seeing that it's becoming part of your sustainability aspects in your grants. Automation is becoming a very important part of the future as well. So that also is fueled by standardization. As we go open more and more, there's an increasing push for standardization with things like AI, deep learning, machine learning, you name it. It all requires and is mounted on standardization efforts. So what exactly is standardization? Well, what I'll do is I'll start by introducing the various speakers that we have here. First up would be Satrajit Ghosh. He actually is going to answer some of those questions, like what is standardization? And, if I'm not mistaken, he has an office down the hall from Tim Berners-Lee, who, is the inventor of the web and probably knows a thing or two about standardization. So I'm sure they've had interesting conversations. Before Satra comes up, I also will mention that Anita Bandrowski is going to be another speaker. She's from SciCrunch. I've crossed paths with her on INCF committees, and so she will tell you all about RRIDs, and there's a whole world there as well. And our final speaker is Guiomar Niso. I've actually had the pleasure to work with her at the Montreal Neurological Institute when she was doing her postdoc there. She's now at the Polytechnic University in Madrid. And so she's going to tell you a little bit about an initiative called BIDS, which has, over the last few years, definitely swept across the neuroscience community. So with that, I think we're at the place where I can call on Satra to tell us everything he knows about standardization in about 20 minutes or so. So Satra?
Hello, folks. Can everybody hear me?
Yes, we can.
Wonderful. And you can see the screen.
We can as well.
Wonderful. So I was asked to talk about this topic, what constitutes a good standard for neuroscience? And the short answer is, I'm not completely sure. But I hope that over the next 15 to 20 minutes or so, I give a little bit of a flavor for the kinds of things you would consider in thinking about a standard for neuroscience. And perhaps the best place to start with this is to think about neuroscience itself. Neuroscience, much like most scientific domains, is fairly complex. As one can see across these different scales of neuroscience, we cover a vast array of species, subdomains, and modalities. And then if you think about the experiments, we collect data from a plethora of devices. And these experiments not only are about data collection, they encapsulate scientific thinking. You build those experience on the results of other experience. Today, you look at all kinds of computational approaches and consider many elements of decision making as part of running those experiments. And this broad space of neuroscience involves theories. You're thinking about models of behavior, computational models. They involve these experimental designs. How do you elicit certain kinds of behavior, certain kinds of observations? Many of our questions are driven by various applications, and some of this are how do we translate neuroscientific knowledge to the clinic? How do we help humans who have different kinds of neurological or neuropsychiatric disorders? We also kind of look at behavioral aspects as drivers. We want to enhance certain proficiencies, deal with certain elements of cognition, and those help us also to think about how we gather knowledge in the space of neuroscience. Because to drive these applications, we want to be able to do the fundamental research as well that drives knowledge, which drives the applications. Finally, to kind of do all of these things, you need tools. You not only need the instruments, which I mentioned, there are various protocols. And across this space of neuroscience, there's a lot of protocols in play. And then as we are moving closer and closer to a almost digital neuroscientific domain, there are various analytics, and these could stem from various pre-processing methods all the way up to statistics, machine learning, or other types of approaches that are done. And neuroscience wouldn't be complete without a rich set of data that's coming online, especially in this digital domain, with rich descriptors, provenance, and other information. An immediate question is, how do we facilitate communicating and integrating information across such a complex system? When we hear the word communication, we think of languages. We have many languages in neuroscience. Every single box over here has its own community, so to speak, its own problems and its own links to other communities. Thus, many languages and dialects exist. The problem is that much like the real world, there's a lot of diversity and crosstalk. It is in such diversity that standards are born. And from here on out, when I say neuroscience, I will refer to this diverse, exciting, and large space of exploration and activities. So standards and best practices help us with this communication. They help us with disseminating information, searching for information, visualizing information, doing computation on information. They also help with collaboration and coordination, and they increase the fairness and efficiency of the scientific process itself. However, the use of standards in neuroscience is relatively young, and the field is maturing as we speak on how to adapt to them. Thus, standards proliferate because we often live our scientific lives in isolation, focused on our own questions, our own neuroscientific communities, and our own implicit knowledge spheres. This does not, however, mean that we do not use standards. We tend to standardize a lot of things within our labs because it makes our lab processes efficient. However, since the dominant currency is still the PDF of the publication, not necessarily the data and the resources or the protocols or the metadata or the provenance of the experiment, our within lab standardization is generally sufficient for that particular currency. So we don't think about standardizing across labs as easily as we think about standardizing within labs. Now, that is all changing. As you know, various data archives are coming online, various places are publishing protocols, and as our community moves towards greater sharing and reproducibility, these standards need to evolve to meet those of others. However, we kind of start in this space. This is a simple illustration of how standards are born. I sent you data. Thanks. This is a Word document containing an embedded photo you took off your screen with a spreadsheet open. Yay, does your computer not support .norm files? Maybe you need to update. And since everybody sends stuff like this anyway, we should just formalize it as a standard. And as a result, standards proliferate mostly out of good intentions to improve the world around us and the lack of resources to coordinate. And as a result, we get this. There are 14 competing standards. 14, that's ridiculous. We need one standard to cover all of these use cases. And now there are 15 competing standards. Dum la resto decitio parolado mi tractos. Kio estes normo, ĉu ni vere bezonas normoin, kio consistigas bonan normon. Now, if standardization were easy, what I just said would make complete sense to you and me. After all, this was constructed and proposed as a universal secondary language in 1887. But while you and I may not be able to understand what is being said, there are certain elements of standardization within this itself. I was able to read and pronounce these things, and that's because they follow certain rules. And if I were able to spend some time with it, I may be able to understand that this translates to, for the rest of this talk, I will cover what is a standard, do we really need standards, what constitutes a good standard. And so let me cover the rest of the talk, and hopefully all of us can learn some Esperanto at some point in time in our lives. So according to the European Committee for Standardization, a standard is a technical document designed to be used as a rule, guideline, or definition. It is a consensus-built, repeatable way of doing something. According to the International Organization for Standardization, also known as ISO, an international standard provides rules, guidelines, or characteristics for activities or for their results aimed at achieving the optimal degree of order in a given context. And then according to the National Institute of Standards and Technology, standards allow technology to work seamlessly and establish trust so that markets can operate smoothly. And what we can do is pull out a few key elements from these descriptions. You will realize that while they're using slightly different words, they kind of overlap in conceptual understanding quite well across these three different agencies. So I'm going to pull out these words: technical document, a rule, guideline, or definition, consensus built, repeatable way, again, rules, guidelines, or characteristics, optimum degree or order, work seamlessly, establish trust, operate smoothly. So what is a standard? Well, first off, it's a technical document, and that's important to remember. It's not an implementation. It's not a specific implementation or multiple implementations. It's a technical document that provides rules, guidance, or a specification. It's consensus built, as in multiple people have come to agree that that's a good technical document. And it helps towards repeatability, creating order, simplifies life instead of chaos, seamless and smooth operation. And perhaps one of the things that's important for many of us in this community is it establishes trust. Now, standard building benefits from coordination. And for any funders listening to this, I would suggest please think about how to support coordination efforts that help with developing standards that work for many people. And the process of creating a standard may itself be standardized. On the right panel over here, I'm showing some of the standards that the INCF has supported and as a standards organization are starting to endorse or going through the endorsement process, which is a process that goes from submitting to expert review to community review to committee review and then an endorsement of the standard. But do we really need standards? Or do standards get in the way of research? What do we really need? So let's start with something that's outside of neuroscience. And this is from, again, the European Committee for Standardization. Can you imagine a Europe with different credit card sizes, a Europe where you couldn't be certain that petrol was the same in every country, or where genetically modified organisms in food were classified differently? A Europe like that would be chaotic for all of its citizens, and daily life would be far more complicated. But many might say science is unlike other forms of standardization. Science is about exploration. So let's consider a couple of examples from the scientific side. Here's an example of a neurophysiology workflow, the NeuroPixels workflow. And many neuroscience experiments are like this. They have many stages to them. They involve many steps and sub-steps. And each process has various inputs, outputs, and various agents who interact as part of each process. And if you look at just this workflow itself, you can start adding up the weeks, and it would roughly amount to about 3 weeks to a month of time over which this workflow took place. Now imagine the final result that comes out of it is some claim made in literature. That claim would rely on all the components that went into this workflow. And to make that claim robustly, one would need to understand that the various elements of this workflow were fairly standardized so that it could be reproducible. But some elements of this workflow also involve human interaction. So here's an example of one of the steps on just evaluating the quality of a process. So this is the intrinsic signal imaging or ISI quality control process, and that itself is a process, something we might want to think about standardizing over. I can switch gears a little bit from neurophysiology to neuroimaging. And if you think about neuroimaging, the entire life cycle of neuroimaging is also a process that involves many different components. There are components involved with subject and experiment information, there are acquisition details, there are ways to store and curate raw data, and you'll hear a lot more from Guiomar on this. There are analyses that go on, there is derived data created, and finally we get to publications and dissemination of information. And this process is not necessarily linear, it may be iterative. An intent of standardization is to move away from this focus we have on publications to a focus on the process so that we can understand the components of the process and effectively reflect them in the publication. And different stakeholders in a domain may have different needs of viewing on this process. So one needs to understand standardization from the context of various different community members who are looking at this process in different ways. And this process in neuroscience involves standardization over many different components: data, metadata, provenance, protocols, computational processes, other interactive processes like quality control that people do, various software, various services. They could be compute services like APIs or database services, and then computational models or prediction models. Neuroscience also involves various biological and chemical entities that come into play at different levels. And finally, we need a way of standardizing the knowledge that comes out of neuroscience as well. So I would say there's a strong need to standardize information in neuroscience. Because as the ANSI Institute says, it ensures that products, processes, and systems we rely on every day are safe, reliable, efficient, and work effectively together. So since we can kind of say that standards might be required, what constitutes a good standard? And how do we build such standards? So I'm going to spend a few minutes talking about this, but I'm going to start with something that the INCF Standards and Best Practices Committee published earlier this year. And this is a way of evaluating standards. So let's start with this. A standard should be open. And is it open according to open definition, and does it follow open development practices? It should be considered from the FAIR point of view or relevant FAIR criteria. Is the standards and best practice itself fair? Does it result in the production of fair research objects? Some of these criteria may not apply in all cases, so we have to be careful when thinking about some of these standards and thinking about what applies and how we evaluate them. One of the things within the INCF context is whether there's testing and implementation associated with the standard. If the standard and best practice is supported by appropriate software, is that software open? Is it well designed? Is it implemented, validated, documented, and available for use? Does the standard and best practice have a governance structure that makes it clear how decisions are made and how grievances are handled? A standard is not just the technical document, it's also the community around it that helps maintain that standard. Does the standard and best practice have substantive evidence of use outside of the group or individual that develops and maintains it? So a standard should be broadly used. You can make many guidelines, rules, or suggestions for things that could be, but as a standard, we hope that there is substantive evidence of use outside the group. Stability and support. Who is actively maintaining and supporting the SBP? And what are the plans for long-term sustainability? We'll get into this a little bit more in the next slide. And a comparison with other standards and best practices. As I mentioned earlier, many of us have been living in our own silos. And as a result, there are competing standards that try to solve the same problem. But competing standards add extra burden to the community. How do we think about a process that helps reach consensus across standards, or say that there are two standards that are trying to solve similar problems, but in a complementary way that's useful to the community? You've already seen this part of the slide about what is a standard. So let's go and ask a few questions before you think about creating the next standard or what would turn out to be a good standard. The first question you really want to ask is, do we really need a standard? And you can answer it by answering the following questions. Will it improve interoperability? Or more broadly speaking, all of the FAIR criteria? Will it improve research effectiveness and or efficiency? Will it lower cost? And this is where I'm going to come back to cost later, because we are talking really about the total cost of a standard. And what does the standard depend on? As in, how fragile is the standard if you start building it? The next question is, what is the standard for? And we've already covered this, so I'm not going to go repeat those details. In neuroscience, you're covering standards across a lot of different things. And the complexity of neuroscience has to be taken into account. Does the standard scale? Will it handle the evolution of information and understanding in its domain? However, as a good standard, I think it has to be focused on solving specific problems. That's where you start while asking those questions. Finally, I think the who is as important as the what. Who are the stakeholders? Is a community necessary for developing a standard? Sometimes some standards can be built by a single person. You don't need a community. But what you need a community for is the acceptance of that standard as something that's useful. And here there might be some differences between industrial practices and scientific practices, but in general, you do want to consider your stakeholders. And you want to consider the capabilities of your audience. Is the standard intended for other software developers? Is it intended for technicians? Is it intended for scientists? Who is your audience who will interact with the specification is important. And standard building should depend primarily on the current state of knowledge. A standard building should not be open research, but it should have a view towards what advances in the research space are in the pipeline. You don't want to do standards based on just the past. You want to think about what it is about the future that you need to care about, even without building full capabilities of the standard towards that future. And this is perhaps one of the most important things that I would like to convey about standard development. What resources will it take? This is where you calculate the total cost for different groups. What is the cost of specifying it, bringing all the stakeholders together? What is the cost of maintaining it? The world evolves. And this is perhaps more true in a nascent informatics field like neuroscience, where things are changing rapidly, which means the standard really needs to be maintained. What's the cost of adoption or compliance? It may require education, it may require training, it may require additional resources per lab. So the cost of using it is important to kind of take into account when you're building a standard. And you have to think about the cost of competition if you cannot converge or reach consensus, which would still be most appreciated if people building competing standards can work together. But you must take into account the cost of competing when that's not the case. And this is perhaps the social elephant in the room because it depends on funding, has impact on careers in various ways, and creating a standard does not necessarily get you this kind of credo today that you get from creating a nice publication about scientific work in neuroscience. I would like to say that a good standard is invisible. You don't think about that gas or petrol you fill in from the pump, or the electrical plugs that you're using to plug into your outlets at various places. Most of the time, you don't even think about them at all, what a good standard is. And this involves both an awareness curve and an implementation curve or an adoption curve. This timeline has to be balanced against the changing state of neuroscience. So you want to think about what can I do to create a standard that will be so widely adopted that nobody will even think about it. And finally, who will sustain it? Standard building are not just academic exercises towards publications. They can impact an entire field, multiple labs, and domains. So on one hand, one could say this means that others will help to sustain it, but you want to be able to take into account how standards will be sustained, whether it's by coordinating bodies like the INCF, whether it's by consortia that will sustain it, or whether through funding. You want to think about the impact if funding goes away tomorrow for a standard building process. What will happen to that standard? So I'm going to end with the following. This is from about 13 years back. We will be adopting the best practices in our industry, just like everyone else. If everyone is doing it, best practices is the same thing as mediocre. Stop making mediocrity sound bad. Sorry. This is quite relevant. I mean, in neuroscience, we are always trying to push innovation in every scientific domain. Standards are really about creating consensus across people. So perhaps a different way of thinking about this is the following, that standards reflect the shared values, aspirations, and responsibilities we as a society project upon each other and our world. And with that, I thank you for your attention, and I will take questions, I think, after the next few speakers as part of the panel discussion.
So yeah, thank you, Satra. I have to admit, I've said this before. I always enjoy listening to Satra talk. I feel smarter after the conversation is done. Of course, 20 minutes later, I go back to my usual reality, but that's another story. So as far as questions go, I was thinking we could have one or two now. There was one that was brought up in the other forum. So it was from Tom, and he was asked, Tom Gillespie, I believe is his name. He was asking, can or should a standard be a substitute for community norms? Rather, in the absence of something approaching a community norm, is there anything to standardize?
I think that's a great question. So community norms, I think, are a precursor to standards. I think that the level that you want to hold for a standard is something that everyone will adopt and conform to, and you have implementation over, right? When somebody says, here's a USB-C standard, you want adoption across various people using that standard. Community norms are a great way in building consensus towards a standard. And I think they should absolutely be the precursor towards a standards creation.
Yeah, I totally agree. I mean, that seems to be the way that a lot of standards begin. So that's an important part. I have one quick question, and then if there are no other questions, we'll move on. You mentioned in your talk, Satro, that a standard should be open. Now, I happen to agree, but can you tell me why that is the case? Why should they be open?
So I think when we are talking about standards, there are multiple ways of thinking about standards. Obviously, for some people, creating a closed standard that just works might be a competitive advantage, as long as the other things that are built on it are things that people are looking for. I think in neuroscience, so there the context of standards was really about within the neuroscientific world, where we need trust in all the elements that are implemented as a result of the standard. And there are two things I will say. One is a standard is still a specification, it's not an implementation. So in many cases, that specification can and should be open, especially in the research context where we are still playing with so many variations in the system that we need to understand, can we trust the output of some implementation of a standard? So while we are still exploring that space, I think every standard should be open. Personally, I would want every standard to be open all the time. But I can also see potential commercial advantages for certain types of things, as long as there's rich validation criteria of the implementation of a standard, that what you're expecting from something is what you're getting as a result of having a standard. So the standardization is about a process and a way to validate it. And if we can do that as a community, then perhaps certain things could indeed still be proprietary. And in neuroscience, actually, many things are proprietary in terms of protocols and processes.
I saw a cool podcast with your fellow colleague, Lex Friedman from MIT, and he was talking to Robert Breedlove, and Robert made an interesting point about that there's a cost. So he was talking about open networks, or networks in general, but there is a cost in protecting a closed network that is just not there with open networks. So I think similar principles or analogies could apply. But anyway, we'll leave it there for now. I think there are no more questions right now. So what we're going to do is move on to to some of the community adoption stories. So we have a couple that we've chosen specifically that are very interesting, and we'll kind of move down those one by one. And then at the end, I forgot to mention earlier, we're going to have a panel discussion. So after we go through these community adoption stories, we're going to actually bring this out to a larger group. And I will for sure say at least one thing contentious over there, because that's just what I do. But we'll discuss some of the issues surrounding standardization and where it's going, where it's been. And so that'll be an evolved conversation. So if there's other questions, please feel free to put them in the chat window, and I'll either insert them strategically after the community adoption stories, or during the panel discussion, or at the very end. So with that, I would like to call upon Anita Bandrowski to talk to us about RRIDs and all the value that they hold. This has to do with basically having persistent URLs and identifiers for all kinds of data resources. And with that, I will ask for Anita to come on board. Are you there?
I am here. Can you see? Let's see.
I can see you and I can see your screen. And so welcome
really
aboard.
Good things. So thank you, Satra, so much for that really wonderful discussion earlier. That was, I'll have to call upon your slides here throughout this. But really thinking about this, this is a particular story about RRIDs, and I'm just asking, are these a good standard? And actually, this is currently a question being asked by INCF. So we've gone mostly through the process, or at least partially through the process of becoming an INCF endorsed standard, but we're not done yet. And that has been a very good experience so far, because, again, thinking about some of these questions is really powerful. So I just wanted to kind of remind everyone, or maybe tell some of the people who have not heard this before, but how RRIDs started basically came about in 2012 and 2013, and largely in 2014, we actually started the pilot project. So, and this was really led by both the Neuroscience Information Framework and also the INCF, as well as some of the members of the NIH. And there were a group of meetings which basically culminated in about 25 journal editors-in-chief who began to talk about research reproducibility, and specifically about how animals, antibodies, and software tools would be able to be actually described in the journal article. So again, not data, but tools and resources. And so we actually had a wonderful sponsorship by INCF, a dinner at the Commander's Palace, which was at the Society for Neuroscience meeting. And this was great because it turns out that if you pay for a fancy dinner, most even editors-in-chief actually show up, which was nice. The 2-day workshop at NIH also had people showing up, which was good. And INCF actually provided meeting support and food, which was, again, fantastic. Thank you to the EU for being able to do that for us. And then there was a pilot project. So, after all of these meetings, we actually cobbled together a 3-month pilot project where these 25 journals would ask authors to actually provide RRIDs just for 3 months. And of course, you might remember only 2 journals started on time. They all did their own thing. They did it in completely different ways, which was kind of our goal, to make the journals comfortable with whatever way they wanted to implement this, this asking for RRIDs. And just to see what worked, what didn't work, and what they were comfortable with. So we are currently now in our 7th year of this 3-month pilot project, which has been a lot of fun. So again, bringing these questions, and these are really simple questions, deceptively simple, right? These are questions that Marianne Martone actually asked early on in the journal editors. And I think it was very clear that they could not answer these questions, right? So which antibody is used in this paper is a really, really simple question to ask. It's a question that lots and lots of people are asking every day. And then which other papers actually use this antibody? And again, if you can't answer that question, the paper is not reproducible. So this was sort of the impetus behind this standard. So we're also a group. So RRIDs really came out, and they were the brainchild of Marianne Martone, myself, and of course, Jeffrey Greatha. And one of the things that we actually come from the world of search. So we actually were very attuned to understanding the world of search. And the reason that we actually went with this RRID, right, is because it turns out that there was almost nothing in Google Scholar with the abbreviation RRID, which is not the case with something like a top guideline. Top guidelines turns out to be a really kind of a bad search term, but RRID we knew would be a pretty good search string. And so when we now search Google Scholar for just RRID, the number of results here is actually pretty close to the total number of papers, which is not the case for something like TOP or or some of our database abbreviations, which are also genes like RGD. If you search for that, you get huge amount of papers, but it turns out that that's most of those papers are because there's an RGD gene. So it's a unique search string. It won't probably always be unique, but it's a fairly unique search string. We also did a lot of work before we started. So we actually gathered a tremendous amount of data. And these are all of the different data repositories and these are not really just data repositories. These are stock centers. They happen to have data about like flies or plasmids or fish or pigs or rats or lots and lots of mice or antibodies or other things. But all of these places, right? These are all both data repositories, and they are largely repositories for the organisms or the samples themselves. And so there was a tremendous amount of data that was done before we started to actually align all of the data and standardized all of the ways that people would interact with this vast amount of data. And so we created a single index. We put that into a single website. And then over the years, we actually have also created and sought out other bits of data about each of, or about some of these research resources. Some of that is, who's citing any one of these. Another bit is who is telling you anything about this, like which of the antibodies is better, which of the antibodies is worse. And those actually are statements about the RRIDs, and those are all consolidated into a single search portal, which helps people complete the task that we are asking them to complete, which is to go and look up some information and a number, essentially in a long list of numbers, and put that, copy and paste that into their paper. So how were they implemented was an interesting thing. So first of all, we created a lot of different materials for the journals. So again, this was intended to go into the journals. So we started with a set of slides that journals could look at. These are public. We also started with a bunch of draft instructions to authors. But then what I am calling here, and this is just, a set of my major talks over a couple of years. I pulled this out of my my recent file just because it was a kind of an interesting thing, and you're not intended to read this, right? This is not intended to be the case. But see these purple things? These are all the bits of purple here are basically all places where I had actually talked specifically to either one or more journal editors, including, this one, which was the complete editorial board of AAAS, which was, again, very nice. But it took actually almost another year and a half before we actually got movement of RRIDs at AAAS. So it was a massive nagging effort, largely on my part, but certainly not entirely on my part. Almost every one of these talks that I had given in this 2-year span that's represented, I had talked about RRIDs. And many of those talks were to editorial boards. So it actually takes a lot of work. It's not just putting up some slides and giving a position paper. It actually took a tremendous amount of nagging, I will say, of these poor journal editors in chief who actually needed to then go in and do something, right? So it was their job to actually go in and do something. So we also did publish some papers, which was good. First set of papers that we published basically highlighted how RRIDs actually do make more reproducible papers because you could find stuff. So if you ask authors, to actually put up, for example, an antibody identifier here in this bar graph, and you look at the antibodies, whether or not you can actually find them in the paper. This is in the issue of the journal before we started asking for antibody IDs, and then this is the one after, and you could see that there's a huge change here. And so this was actually very compelling to some journals. And we had Neuron, Cell, and eLife really join the initiative after they saw this data. A couple of years later, we went in and did a text mining exercise on the scientific literature. And we looked at that point at the usage of the contaminated cell line. So there's a piece of information here about this cell line, which states that this one is problematic. It's been contaminated. It is a known HeLa derivative. And there's a list of these cell lines, actually, that was put together by an international committee, a consortium to actually try to clean up this literature. And what we wanted to do is we wanted to see, hey, is it possible that RRIDs are actually impacting the usage of these problematic cell lines in some way? And we actually found, after a text mining exercise, that 16% of the totality of the open access literature at that point is actually using at least one of these problematic cell lines sort of in the wild. And the 634 papers at the time that were using RRIDs to identify these cell lines actually, only showed that 5% had a problematic cell line. And again, many of these cell lines are fine to use. You just have to be aware of the particular problem. This is a particularly bad one, but they're not all like this, right? So these were just about only one paper actually used a truly contaminated cell line out of this list of problematic ones. So this is a huge drop in the use of problematic cell lines. And after this paper was published, I just wanted to bring up the fact that we did tell people. And the AACR, which is the American Association for Cancer Research, actually joined the initiative, and they did a whole big thing on their website. A lot of the other cancer journals have joined since then. So going back to this antibody question, again, we did a lot of text mining, and text mining is fantastic. So we were able to look at essentially the percentage of identifiable antibodies in the scientific literature. So this is text mining 2 million papers in the open access subset of PubMed Central, going from 97 to 2019. This was published last year. And what this is measuring, this blue line here, is the percentage of antibody mentions that actually are associated with a catalog number, right? So that catalog number association is about 10 to 20%. It's kind of bobbing up and down here. It is rising slowly. But one of the coolest things that I had ever experienced in looking at any of the data that I had ever seen, coming out of the lab especially, was this inflection point. So here in the 2015-2016 timeframe, where we actually had some of the big journals like Cell, Neuron, and Journal of Neuroscience really start standing on principle to ask authors about, and nag authors again, about RRIDs. What we start to see is this inflection in the overall literature. And so we actually got, over several years of the RRID initiative, this fairly large shift, again, in the overall literature. And we're going to run this again this year to figure out whether this has continued. But this shift is being really driven out of journals like, again, Cell and eLife, which joined. They were bobbing along down here. You could see this. They were more or less in line with the averages here. But you can see that after they joined the initiative, they went from having a relatively average number of catalog numbers associated with their antibodies to having 90% plus catalog numbers and RRIDs, obviously, associated with all their antibodies. So this is in the entire journal, which is very cool. PLOS actually had instructions to authors, so they're actually another RRID journal, but we knew that nobody read those, and since 2015, we have very good evidence for it, and there's more evidence here that, again, nobody reads instructions to authors, because OncoTarget had actually no change in any instructions, and they actually did a little bit better than PLOS. So it was interesting. But the other interesting part is that they are both up in 2019. So there is a sea change kind of coming, it seems, in the best practices of the community, as far as we can tell. Okay, we're part of several standards, including JATS, which is the Journal Article Tagging Suite. And this is the little portion of the JATS standard that actually describes RRIDs. You can see this. There's a Harlan mouse here that's actually been immortalized in this particular standard. We're seeking to become part of a lot of other standards. I'll show you one other standard. So RRIDs are kind of a standard in and of themselves, but they become part of a lot of other standards, including the STAR methods. And of course, we are part of the Materials Design Analysis Reporting Checklist for authors. This is the MDAR reproducibility checklist, which is one of those, 15th standard kind of standards, because there was the STAR methods from Cell Press, and there was the Nature checklist from Nature, and all of those people came together. And actually, they had just published a manuscript on the MDAR reproducibility checklist, but but even though it wasn't published before, science had already started requiring the MDAR checklist. But of course, if you look at the first several items in MDAR, RRIDs are prominently displayed for antibodies, cell lines, and organisms. And so through the implementation of this standard at journals, we actually will have, again, a nagging of a more automated nagging of authors, because each science author must fill out an MDAR checklist. So again, not just being a standard in and of ourselves, but being part of many other standards. Okay, and then of course we've actually written a lot of robots that actually lend a hand. So Tom Gillespie actually wrote SciBot, and this is a representation of SciBot. So here is a portion of a scientific paper, and there are some RRIDs highlighted here in yellow. What SciBot does is it reads the text, and then it produces an annotation in hypothesis. I won't go into how that works, but essentially it takes this, it hits a standard resolver service, and it brings back information for curators to be able to look between the text and the standard information from the database, and to compare those two things. The cool thing is that this is now released relatively quickly to public. And because it's released to public, we actually have another robot which picks up that public information. And this is, again, at CC0. It gets picked up by Crossref and others. But this information is also now tweeted, which goes back to alert the authors of this particular paper that, in fact, someone or something has thanked them for essentially doing this task. And this can happen very, very quickly. As long as the paper is open, we can get a tweet out, automated tweet out, in about 2 days. So that's been very cool. And these things do get retweeted quite a bit, which is interesting. So again, thank you for listening. But also, I would be remiss in saying, go on about your merry business without saying, hey, please use RRIDs in your next paper. And of course, use them on all of your web pages. So, and there are way too many people to thank for the RRIDs, including Marianne Martone, Tom, but also all of the massive numbers of journal editors who have been bugged by me and other members of our lab to please put RRIDs into their journals. Okay, thank you.
Hey, thank you very much for that, Anita. I mean, I find this very useful in so many different ways. I'm pretty sure I actually reviewed MDAR in one of some task I had to do. And it's a very useful checklist. So there's all these neat tools and resources that seem to be coming from this. Maybe I'll just ask one question quickly, and then if there's others, feel free to put it in the chat, and we'll maybe take a couple of minutes. But is there any kind of limitation to the kind of information or data that RRIDs could link to? Like in my head, I feel like I could link it to almost anything, right? So, but would it make sense to link it to an entire description of a project versus, I mean, you spoke of antibodies and maybe cell lines, things like that, but what are the limitations? What can it do? Maybe a couple of words on that.
And what can it not do? So for example, it is not a link to data, right? So we have data citation principles and we have DOIs. Those are very, very good for data. RRIDs are not for data. So it is definitely not for data, but it is for the things that are being used. So the whole idea with RRIDs is what are the inputs for the manuscript, for the paper that should be used. And so these are largely things that are covered under the materials and methods sections, but it could also be for things like core facilities. We have had a lot of work recently done with core facility owners who really would like to get credit. And it's very hard to figure out how to thank core facilities. I mean, there are authorships that some of them require or request, but people don't want to do that necessarily. So there are things that are really hard to cite as part of your paper. And so what are the things that you would like to cite, largely in the method section, that help you do that experiment? Those are largely the sorts of things that are covered under RRIDs. However, we must have an authority that covers that kind of thing. We can't, do this out of thin air. There has to be a good database, a set of information about that thing.
Right. So Marianne mentioned that it's not for data, but they are for databases. So sometimes we have that situation. We have a Canadian Open Neuroscience platform that we've built that, you need to link to it in some ways and describe the data sets and things like that. So that is potential use case, but it obviously has very specific use cases. So not for the individual data sets themselves, but a lot of the other things that you mentioned. And Marianne also wanted to say that you didn't mention the help desk.
Yes, the help desk is very, very important. This I did fail to mention that. Thank you, Marianne. We have a very active help desk, and we actually do help people every single day, usually. There are currently about 10 to 20 antibodies being registered every day. There are about 3 to maybe 10 software tools being registered every day. And there's a general help desk that is queried, and lots of people are confused about where they find their RRIDs, that we get a couple of requests daily as well. So it's just a list of numbers, but it becomes so much more than that, because without the assistance from a dedicated staff and without the assistance of the journals, these numbers are just, they're laying around, they're not doing anything. In order to get them implemented and in order to get them used, there is an effort there that needs to be had.
Okay, thank you very much for that. So I think just to keep up with the time that we have, so maybe I can call on Guiomar to talk to us about another community adoption story, a little thing called BIDS. This started actually as an INCF sponsored meeting / hackathon, and has taken off like wildfire. So maybe Guiomar, if you could come on and tell us your experiences, that would be great.
Hi, I will share my screen now.
Sure. Yeah, I see you. I see your screen, so feel free to take us through this.
Everything's fine? Okay. So yeah, as Samir said, just some years ago, the BIDS was INCF endorsed, and I'm going to tell you a bit more about what it is and what's this kind of brain imaging data structure standard. BIDS is a way of storing, organizing, and sharing your neuroimaging data. And why is it needed, as Satra was saying, and as you were saying before? This could be a potential scenario in a lab. Hey, I need to ask Pedro about this old data. There's so much more we can do with it. But meet Pedro, he doesn't longer work in the lab. So Professor Mary will need a lot of help. What's the problem? The problem is data is organized differently between institutions and even within a lab. And this produces optimal use of resources, human resources, because we spend a lot of time trying to retrieve, rearrange the data, infrastructures. Sometimes we have duplicates, we don't really know what's the final data is, so we have data storage space problems, and also financial, because disorganized data has very limited value after the first publication. So on top of all of these, it produces very poor reproducibility of results. So even within the lab where the data are collected. So the solution is to use a standard like BIDS to organize your data. So what does BIDS do? BIDS just describes and organizes your data. So it just organizes it and describes with associated metadata. So it just follows, it's a structure of folders and files that follows a naming convention. For example, you start with your subject number, and then you go with the data type that can be anatomical, functional, or whatever data you have inside. And then you will have your file that has to be named following a convention that starts with the subject. Then you have several entities that you can add, and finally a suffix and an extension. So like this, you have your data organized, but BIDS is more than about your data, it's also about your metadata. So the metadata is organized in sidecar JSON files that are readable by both humans and machines, and that are named in the same way as the data that they are accompanying. So what's inside a JSON file is just a series of keywords with some values in JSON file. And what's the benefit of using BIDS? So for the researchers, it's easy for the researchers to work on your data. For PIs, if you anticipate more than one person will be working on the same data over time, then it's very good to have your data organized according to BIDS. And if you are developing analysis pipelines, there's a growing number of software packages that can read data in BIDS, and it's very easy to write pipelines expecting a particular file organization. In addition, if you ever plan to share your data publicly, and nowadays some journals and funding agencies require it, it's really easy to curate the process. Well, and this contributes to having your data more fair. And also BIDS offers a validator, has a validator tool that can check the integrity of your dataset and help to easily spot missing values. So should I use BIDS? If this is how you feel when you are trying to work with data that was acquired by someone else, or even worse, by you a long time ago, then BIDS is for you. So welcome to the Marie Kondo of neuroimaging data. The principles behind BIDS is, the first one is adoption. As Satra was saying, one of the key aspects of any standard is to being able to be adopted by the most people as possible. So adoption is crucial. We want to minimize complexity and facilitate adoption. For this, we don't want to reinvent the wheel. We want to use and reuse existing methods and technologies whenever possible. Then we try to tackle the 80-20 rule, tackling 80% of the most commonly used cases. And finally, we try always to engage the global neuroimaging community, especially during the creation of the specification. So if everyone is involved, then it's more likely that more people will be happy with it and will contribute sharing their ideas. So it has three pillars. Like, first one is the standard, which is the technical specification, as Satra was mentioning. There's a technical specification that's the standard. Then we have some tools and that facilitates the adoption of the standard and working with data that follows it. And finally, the BIDS community and the collaboration. For the standard, the specification, we have just released version 1.6 that has merged PET. So now PET neuroimaging data is already available there. And here you can find all the details about the keywords, about the specifications your data has to have. So for example, if this is your data, you will have your dataset description, participants, and then your subjects. If your dataset will have a JSON file that has all these keywords to store the associated metadata. Your participants, the same. You will have a TSV with all this data and then a JSON file with the descriptions of each of the fields. If you have MRI data, you will have a T1 weighted JSON file that could look like this with modality, magnetic field strength, imaging frequency, manufacturer, all these details. For MEG data, this will be equivalent, the same, about the power line frequencies, sampling frequency, MEG channel count, EEG. So all these fields that has to be stored in the sidecar JSON files are on this documentation. Some are required, some are recommended, and some are optional. So it's up to you how the level of detail you want to include, but there are a minimum of things that should be there, always there. So the bits started for MRI at Stanford at Ras Potrak Lab with Chris Gorgolevsky, and for MRI, and yeah, following the Open fMRI that now is Open Neuro database, they found that they needed to create a standard to organize the data. And since then, MEG has joined, and we extended the specification for magnetoencephalography data, also for EEG and for intracranial, and now many more extensions has joined. Like the last ones were the arterial spin leveling, and very, very recently the PET. So this is very good news, and we are trying to broaden the community and to include more modalities, as Satra was showing in the first image. There's like so many things. The neuroimaging community is so big that and refers to all these levels of modalities and specifications that it's very difficult, but we are reaching a very high number of them. And this is all the extension proposals that are work in progress. So we have very recently added microscopy and animal electrophysiology, and in addition to all of these that are already being working there. So if you want to join the effort to some of these initiatives, please join. And if you want to propose a new one, you are completely free to do so. So just put some energy and contact us. So BITS tools. Multiple tools exist to make it easy for researchers to incorporate BITS to their current workflows and to maximize data sharing opportunities. Like BITS converters, for different techniques like MEG, EEG, and MRI, and different, and also general tools for data querying and data plumbing operations like PyBits and BITS MATLAB. And finally, BITS apps, which are containers for analysis pipelines using Docker or Singularity. This facilitate the analysis in neuroimaging and reproducibility. Also, we have a validator to check the datasets. And it will help you to spot some missing values or something. It will give you some warnings if something is not very accurate or something is missing. And finally, the BITS collaboration pillar, which is the BITS community. As we said, BITS is a community-led standard. And like so, it's an ongoing collaboration of a lot of people. So the specification is created sharing knowledge and discussions and consensus through the email discussion list, through shared Google Docs, and through GitHub. So we have also a very well specified governance structure. And BITS extension proposals are called webs, and each of them has a leader and a place where people can go to if they want to contribute easily and know the channels, and also a BITS steering group and a BITS maintainers. This is the steering group that was elected, and we are people from all around the world with expertise in different neuroimaging modalities, which make us a bit with a broader view. And there's an awesome team of BITS maintainers that really put a lot of effort and energy to make this standard to move forward. So BITS is very open and inclusive community. There are several hundreds of people contributing almost daily on GitHub, and you are really welcome to come and contribute. Contributions could be made at all levels. They could be low level, intermediate, or more expertise. And so you are completely free to join and contribute at the level you are at. And we have a BITS starter kit here that people can, that doesn't know BITS, can get on board. And yeah, BITS is changing the way in which neuroimaging research is being done, and it's been adopted around the world and with many contributions. So a global community of over 300 people created BITS standards and tools for neuroimaging modalities, and more to come. So BITS is really good for future users of your data, but also good for future use. So keep your data organized in BITS. And thanks to INCF for endorsing the standard, and to all of the contributors.
Okay, thank you very much. That was nicely outlined, in terms of all the different components of BIDS. Maybe I could just ask one quick question before we move on to the panel discussion. So what do you think is most missing in terms of the next steps with BIDS? I mean, you listed all kinds of different modalities that are starting to standardize using BIDS. What about maybe the derivatives? Do you have anything to say specifically about derived data for each of those files, or what's next, basically?
Yeah, I think that the derivatives is like the next step to go. There's been some effort that has been done, and the common derivatives has been released, which is the minimal definitions and minimum set of things that needs to be ready before starting to plug in some derivatives. And there's ongoing work on different kind of derivatives. I'm personally involved in the electrophysiology derivatives, which involve MEG, EEG, and intracranial EEG. So I think this is the next step, because now we have get the data from the scanner to a database or to our place, but now we need applications and need to process this data, and we need to store this data. And for this, we need a standard, and we need that the standard gets extended.
Okay. Is there any suggestions on how to move forward with BIDS derivatives to get the next level of derivatives done? Should INCF pay for all of it, or how does this work?
Well, yeah, help is always welcome, and support always welcome, because this is something we are doing all of from our free time and a side of project for most of us. So yes, support will always be welcome.
Okay. So thank you for that. I don't know if there's any other questions directly on point. BIDS, Matthew is saying BIDS is an example of what can be done through INCF working group. To learn more, just visit the INCF working group booth. So shout out to the booth. You can definitely find more. INCF has been, in my opinion, very beneficial in bringing the community together in so many ways, whether it's the standards or just all the training aspects. Like there's so much that is of value, I think, that the neuroscience community has benefited from. So, I just wanted to mention that. All right, so I think if I could call upon everyone else now to open their cameras, what we'll do is we'll start a panel discussion. And so I guess, should I start with something more contentious, or should I start nice and easy? I'm debating that in my head right now. Maybe I'll start with a nice, easier type of question. So to the general panel, what would you say are the biggest challenges in neuroscience standardization today, right? Like, so, we've heard from bids, and some of the things that need to be done, whether it's derivatives. Like, where does the attention need to be right now? I mean, there's standardization efforts all over the place. So anyone have an opinion of where this might be, more attention might be needed, some immediate, action? So I'll call names if you guys don't pipe up, but feel free. Yeah.
Can I join in? I think that one of the places which is of immediate sort of some immediate attention needs to be paid is a lot of the closed formats for a lot of data would be one of the places where some real good can come out of a group like this, right? If a lot of us are actually talking about closed formats, getting rid of some of these closed proprietary formats, it would be really quite amazing. And really talking to the companies and with the companies that provide a lot of the tools that many of us use. I know that with bids and with some of the other standards, we've gone a long ways towards that, but I think a lot more needs to be done, especially
Is there a specific domain that you're thinking of more than others, like a type of modality, perhaps?
EFIS would be great. That's the one we're looking at right now.
Noted. I believe Liuba, you had a question?
Yeah, a question or a comment. So I was wondering, because you asked where standards are needed, I have the impression that especially the areas where data sharing is actually not that popular yet and just begins to start, these are the fields which lack the most standards in general. So at least I have the feeling. So my question would there be, isn't that a sign that standards are typically lacked where data sharing is not yet really frequently done? And neuroscience in particular, electrophysiology is one of the big areas where data sharing is, for example, not very common yet.
I was waiting for someone to bring up electrophysiology. There has been some recent pushes, like with EEG bids and things like that, but that was the Wild West not too long ago, and maybe to some degree still. So that's definitely an area that people have started to realize that there needs to be standardization. Agreed. Any other comments about that? Any other areas that...
I'll make one comment about electrophysiology. So I think one of the issues there is that there's a lot of times metadata that's kind of sort of specific to a lab that maybe should be included in the data set to make it really, to make the data set complete. But then there's effort involved in representing that in a standard way. And so the standards that have, at least that I'm familiar with, NWB1 and NWB2. So NWB1 allowed people to put kind of custom metadata into their file, and it was just labeled as custom. So it was something that was in the file, but it was not part of a standard. And NWB2 did away with that, as far as I know, and it requires everything to be specified by an extension, which gives us a schema for additional metadata. But then that requires work on part of the experimenter to design the extension. So there's more work there. And so I think part of the issue might be just the complexity with electrophysiology data, just that it's some of the data is hard to really standardize, and there's more work.
Yeah, we've noticed that with whether you have the electrodes themselves that need to be described, or the actual the signal files, they also have there's so many different parts of electrophysiology depending on whether you're an EEG or not. But yeah, that's definitely a challenge. So I guess, but there's still great value in coming together so that you can interchange the data from one group to another. So definitely. David, do you have a comment?
Yeah, So the next frontier, in my view, for standardization is metabolomics and proteomics. You know, what's in your blood. These has a lot of promise for personalized medicine going forward, and it's really the Wild West, even down to analysis methods. Nobody knows how to correct for inner site differences and so forth. So.
Okay, yeah, I totally agree. There's a whole slew of actual data types that need to be standardized. That's definitely true. We've heard some examples there. There's also other aspects of what we do with the data that needs to be standardized. Like, for example, I work a lot in high performance computing and pipelines and all that kind of mumbo jumbo. So standardizing APIs, right? That's very important. Imagine trying to get your data off your Apple Watch. Well, sometimes that's not as easy as you might think because there's no way to do that. Or containerizing your pipelines. For example, I'll shout out to my colleague Tristan Glatal, who came up with something called boutiques, which is a way to describe the pipeline that you have. If you've installed pipelines enough times, you start to realize there are certain patterns to that. So even in those kind of areas, there are things that need to be done. Liuba, a comment perhaps.
Yeah, I wanted to ask if, I mean, maybe all of you have the same experience, but coming back to the question of what is a good standard. So for me, I rather would not like to invent another standard, but more connect to the ones that are already present, but identifying the ones that are actually the ones that gonna go through. And like, I mean, BITS was a very obvious one, for example, that we are connecting to our RRIDs as well, but there are a couple of others outside there which I think are harder to identify. So just recently I struggled with strains. So what's the standard for strain names? There are so many out there and conventions, and apparently not a very good agreement on one. So I rather would like to learn about ideas from your sides in order to connect to standards, to make actually use of them and interconnect them with each other, because they're covering different and specific fields, right?
Agreed. Maybe this is a good time to interject a little question from, I think, the audience. Jordi Huguet, I believe is how you say that. So the question is, bearing in mind that most neuroimaging data is nowadays acquired and exported from modalities using DICOM format, isn't an extra burden to take care of and add an additional step to convert it to BIDs? What are your opinions on that? Anyone? Maybe Satra, perhaps, since I see your hand raised.
Sure. I was going to start with answering something about Liuba's strain thing and point to Lydia and say she has a lot to talk about strains and genotyping if you want to get details. There were two things. Can I start with that previous thread,
yeah,
and then I'll answer Jordi's question?
Whatever you want.
So one of the things that I will say is kind of going back to the question Tom had asked after my talk about community norms. And I think what we are seeing in neuroscience is a lot of things are evolving fairly rapidly, and it really requires kind of pieces of the community before we even talk about a standard to kind of agree on what makes sense for them. I think standard is too hard a boundary, or I think it should be a hard enough boundary that when you cross that, you expect everybody in the community to follow. So prior to that, I would expect kind of community norms, and there's way too many places in neuroscience where we don't even have community norms, and we should be working towards that. So that's kind of the focus on Liuba's train of thought. In terms of DICOMs, so DICOM, I was going to have an entire section on DICOM as a standard, and I took that out of my talk. DICOM has been super beneficial to the neuroimaging community. So the part of the reason we could move towards standardization was because of DICOM's existence, but it also leads to what were the problems with DICOMs that led to the evolution of Analyze, NIFT, and then the BIDS structure that kind of built on additional metadata on top of DICOMs. Part of the problem is DICOM is a standard, but getting changes accepted into DICOM is hard. And as neuroimaging started evolving with more specialization of metadata, scanner manufacturers or sequence manufacturers or sequence producers started putting a lot of that into private data fields, thus not harmonized across the scanners that were there. For a long time, Philips produced things in PAREC format, not DICOM. So there was differences across the major instruments that produce things. I think the advantage is that there are a common set of scanners that produce DICOM now, and that's a great place to start. The problem is they often do not have common places for the metadata that's needed to interpret the DICOMs. And a big chunk of what BIDS has done is to normalize that metadata across manufacturers into a consistent manner. So yes, we would, if DICOM had managed to normalize that metadata at a fast enough moving pace as the community that was using it, we would have probably not moved away from DICOMs to start with. But there is a change. DICOM supports all imaging, not just neuroimaging. And that makes it harder for that standard to move or change as a result of it. So I don't think it's, I can't blame either community for the lack thereof or the non-adoption or the use thereof. I think it's just differences in the nature of what that standard supports and how difficult that is to change for a specific, very small subset relative to the world of DICOM, the neuroimaging community.
Yeah, I mean, I agree with you. I have also had a lot of problems with DICOM conversions one way or another and still do. So the question is, how do we, I mean, in the absence of coming up with a standard, whether DICOM or, now we have BIDS, but it seems like we're still going to have to spend a lot of time on conversions, right? That they have to be maintained every time there's a software upgrade for the scanner, different scanners, different software upgrades. They have different header maps and all these things that you need to do to keep up with. That requires a lot of attention and work. And if it's a voluntary initiative by the community, that just, I don't know. I feel like that has a sort of a limitation. So any comments to that?
Yeah, I can jump in here because I think this is something that it comes up repeatedly. It's coming up in electrophysiology now, where there's many different formats. Many of them are proprietary, and the question is, should we write a whole bunch of converters and maintain those as a community, or should we try to work with manufacturers to get something like DICOM for the electrophysiology community? And there's a balance here, which is you say, who is this standard for? I think Satra brought this up. There's an audience for these standards, and so DICOM and many of the pushes to get NWB adopted by manufacturers, those are where there's an audience for that standard, which is somebody who's making a tool, and they have very different timescales, different requirements. I mean, I don't even want to imagine how far in advance you have to plan out a new line of scanner, right? That's decades worth of work. So they operate on radically different timescales than we do in academia. And so if we want to have standards that are able to be extended and open by the community, you wind up with something like BIDS, where the audience is the people who are making and writing the pipelines. And compare that to somebody who, I say, we should standardize how to communicate protocols to each other. But the problem there is, who's the audience for a standard way of doing that? That's every single scientist on the face of the earth. Right? There's no way that audience is going to be able to come to some community norm, right? Which is sort of a prerequisite for being able to standardize anything.
Yeah, absolutely.
Can I bring up something quickly? I'm going to give Guiomar a chance to talk about this, because I think this was part of the discussion that happened with the BIDS MEG extension of the different file formats that were supported under it, because it's not just getting to a standard, it's the downstream part of it as well. So it's about how data converted to that format is used. And I think during that process, during that BEP, there were discussions of trying to get to a single format, and that went away because of the lack of resources that would apply to changing all the instrumentation post-acquisition into that format in terms of analysis.
So Guiomar, you can say a few words perhaps to address that. If not, I have all sorts of stuff that I'm happy to talk about regarding that point.
Yeah, just very briefly, that Satra is completely correct that because in MEG, we didn't have a DICOM format to overall everything. Then we have so many different formats. What should we do? Shall we just condense them into the most used one? And because we wanted to be inclusive, and there were many hard discussions about where to go, and finally we really decided to keep it open and respect the history of each of them. And even though it's maybe less efficient, but at least everyone could feel represented and could feel more involved in the adoption of the standard.
Yeah, I mean, that's a good answer. Definitely, it's good to try to. So some of the issues is, how do you bring everyone together? You know, it's good to keep it open. It's good. I think the initiative of BIDS has done a very nice job of, you know, educating and training people and having open discussions about things like that. So that's definitely needed. But what do you do if you have, say, two competing standards or formats? I don't know if I want to get into exactly Mink versus Nifty, but what happens if, you know, you have a situation where there's these two competitions and one of them is maybe better than the other, and then it takes a life of its own? So do anyone have any comments about that? What do we do? I could keep talking about Mink and Nifty. I mean, just to give you a little more, you know, like fuel to the fire. So Nifty had some limitations, right? In terms of, it was a format. I think people were thinking about it in the way of a standard in many ways. Large part of the tools were adopting it. But it obviously had many limitations. Would you like to know exactly? I can quantify that. Everything in BIDS is what was limited about Nifty, right? So how do we deal with situations where there's multiple standards? Liuba, I see your hand up.
I found this a bit like, I mean, the Mink Nifty situation, I'm also aware of. We discussed about this in the past INCF meeting a lot. So from my perspective, it's really a bit like, I mean, the Nifty Mink discussion sounds to me, which I'm coming from electrophysiology and really from animal electrophysiology, the discussion between two formats sounds for me like, well, I would like to have that situation. And only that situation in general. So I don't know, I think considering BIDS decided to support Nifty now, in principle, the structure, right, as they do for MEG or for IEEG, they support also multiple formats. It wouldn't make, I think the important part about BIDS is rather the structure and the accompanying metadata that are more formalized in a way to make it really successful. The format itself just needs to be readable by some open source tool, right? And then we're coming back to FAIR in sense of, well, there could be multiple formats out as long as you can read them. And if you have the corresponding metadata in order to navigate through the data, the consistency in a data repository, I think the actual format, for example, the standardization there doesn't make such a big deal. I mean, it's nice if you only have one, but from my perspective, the format discussions are the least bit of a problem from my perspective.
I think that there is also, if we think back to Ken Harris's talk about the fact that, okay, we want a standard way to record electrode locations across a bunch of different brains on different systems in different labs and get them all into the same area. Maybe they were doing their recordings with different software, but the thing is that they had, as a community, a need to do this. So I think that there's another part of this, which is in communities where we see that there are multiple different competing candidates for standards, it's not clear that there is necessarily a use case for data integration that is being inhibited by lack of a standard. And so finding a balance between you know, it's like, what do we gain by trying to normalize in a community if there's not an impetus for this unified integration? You know, is anybody funded to integrate data that is being collected by all of these different labs? And this is something that we've encountered in Spark, where right now, the key thing that we need is people using the data and trying to understand that. And then we'll know whether there are cases where we want to integrate and can standardize.
Ben, I believe you have your hand up.
Hi, everyone. Late to the conversation, but I've been listening in. Actually, I didn't realize I was invited until just now. But so yeah, I actually completely agree with what Luba and Tom are saying. And in the context of Neurodata Without Borders, we see the same thing. You know, where the IBL is using their own format, as Tom said, because they have their own specific use cases. And it's extra performant for precisely what they need to do, whereas NWB more emphasizes flexibility and the ability to handle lots of different data types. And I also agree with Luba that actually that situation is a really good situation for us, because the alternative is 27 different formats. And so if we can build one conversion pipeline, that's like a tractable task. So I really love seeing data standardization, even if it's not Neurodata Without Borders. I love seeing any efforts of data standardization in neurophysiology. But I think it is also worth taking a step back. And on the one hand, you know, they have different communities, and so they end up having this kind of divergent evolution where ecosystems are built around these formats that cater to the communities that are using the formats. And so they end up kind of having different types of tool sets, and they fill different niches. So on the one hand, I think, you know, they start out with slightly different purposes, and they end up kind of diverging even more. But at the same time, it's worth taking a step back and saying, okay, well, in what situations, as these formats evolve, maybe they will converge as well. And it's worth thinking about, you know, building efficient conversion pipelines that allow you to access the tools that have been growing in the other tool ecosystem.
Satra?
So I wanted to kind of bring in a couple of things from my talk. One is the cost of doing something. And I think that's a huge cost for a lab to move away from things that they are doing to something different, even if that different thing is what the rest of the community is starting to call a standard. And I think that's something that we absolutely have to take into account, and how to facilitate that process. And that brings me to the second piece, which is we need, I think, a separation of information and data formats. I think what we can standardize on is that we want to collect information that's harmonized in some way across things. The data formats are going to continue to evolve as the scale and nature of neuroscientific data changes, because the technology is now linked to analytics. And then the third thing that we need is analytics themselves. A lot of it is currently the Wild West. Everybody does all kinds of things. And I think one of the biggest reasons for adoption of BIDs was standardization at the analytics level and the conversion level. If those two things hadn't standardized, it would be much harder for people to move to BIDs than having those toolkits available. So those are kind of three components that, if we're talking, and I completely agree with both Luba, and if it's a competition between two formats, I can live with that. Generally, in most cases, it's a competition between many, many more different things.
Anita, next comment.
Okay, so really quickly, I just wanted to quickly comment on, you know, the fact that it is the Wild West. And in fact, there's even in places where we from neuroscience might think things are settled, they're really not always settled. You know, going back to the strains discussion, there are actually a lot of different competing ways to talk about a strain of an animal, and some of them are ontology, some of them are actually driven out of the genomics community, but the genomics community is not unified, where you talk about a strain of a worm, which is completely differently ordered than the strain of a mouse, than the strain of another animal. And so there are so many different ways to talk about that. So, you know, again, one of the things that we had done is we said, okay, you talk about that any way you want, and we're just going to, you know, stick with the number, which is really easy for people to actually cite and reference. And they actually know that because they go and order that number, right? Oh, that's the 664 mouse from Jack's. Okay, that's fine. Whatever the heck it's called, it doesn't matter. It's the number that actually ends up being the relevant piece of information. Just this is a little bit different. So in one sense, I keep thinking about this. A lot of the things that Satra had brought up and others have brought up on this is the simple thing ends up winning, which I kind of want to state, like we have tons of these different kinds of standards, and it tends to be that the one that's relatively simple is actually going to be the one that ends up.
So Lydia and Jeff, I'll get to you in just a second, but I have a quick intervening comment. So yeah, I totally agree with simple is very effective. But the problem is sometimes some issues require far deeper thought many levels down the road. Otherwise, you have this exact problem like with BIDs, where there was all these limitations with the file format that people were using. So then they added these other layers. And in fact, I think something like BIDs apps is actually what's going to drive a lot of the development, because that is tangibly giving the researchers what they actually need, right? Like the tools they need. So most researchers don't care about the format. That's not a big deal. It's a means to an end in terms of like being able to do what they want to do. So anyway, that's some food for thought in there. I don't know if I totally diverted Lydia's comment, but feel free to pipe in.
Hi, I was following Anita's one in that there are going to be lots of different file formats. There's going to be different ways of doing it, we're in a happy place if people think about standardizing at all. I think the next step for standardizing is, as they standardize, think about the concept they're about to ask people to represent, and then define that somewhere. I think there needs to be documentation of the standards such that, what is it? Is this a strain? Do we mean just a population, or do we mean the allele? If you mean something that carried an allele, then you say so. And then this is where you can call them an ontology, but those interrelationship between things, even if we don't standardize now, but as every data standard starts to define those, as things start to grow, as we merge standards together, you will know which terms go with each other. So beyond where to put the little digital bits, I think it's important that we define what each of the fields mean, and making really sure that it's interpreted in a certain way. So I think it's bringing ontologies in, but not bringing the world's biggest ontologies, even just defining each of the terms that you use in the standards, so that things can evolve.
Yeah, there are initiatives trying to do that. Now, my personal opinion is that trying to ontologically standardize the world is far, far, far in the future. But there are cleavages and areas that you can do, and there are things being done by even people in this group. So I think, but then that's one way of standardizing on ontologies. There's so many others. There's with programming and the data itself and a million other things. But good points. Jeff, your hand is raised.
Yeah, I actually wanted to ask Satra a question about a point that he made on one of his slides, and that was you mentioned the phrase standardized knowledge as a goal. This ties in to what the current discussion is now, and so I'm wondering if you could. So to me, knowledge seems kind of amorphous. It seems like there could be different ways of defining knowledge. And
well, I have an answer to that. Just listen to whatever Satra says, and we'll standardize around him. How does that sound?
Great question, Jeff. So I think it's a hard place to standardize, but I think we can start simple again. Every paper that is published produces some claims. We can standardize that level of claims associated with a publication. And if we can do that, imagine how much easier search would be when you're looking for information associated with a claim. So I'm not talking about the general title or the abstract that the paper puts out. I'm talking about the specific claim that the paper is making, which is buried deep typically. But if that was extracted by the author as part of the publication stream, we would be in a very different place in terms of search today than we are.
But how can one represent a claim? Like how can one standardize the representation of a claim?
So that's another good question. And there are actually conferences devoted to knowledge representation. So I won't go into the details of some of those things, but there are ways and structures in which you can represent structural attributes of a claim connected to provenance, connected to the details of the paper or experiments that one can do. Happy to provide some links to what people have done, but this is a very large area of research that many people are doing. So we can leverage that even in neuroscience to say, this is how we represent a claim. A classical example that I know of comes from work done in Alzheimer's research. So there was a consortium, maybe that's too strong a word, but a group that was created called ALS-SWAN, which represents claims made in Alzheimer's research. And so that would at least be a connected place to start looking at how people were representing those things. The problem is the curation part. And this is where perhaps sticks from publishers or funders could come in, is you make submitting those claims be part of a publication submission or your RPRR as part of your grant renewal documentation. And then suddenly things become quite a lot more standardized in terms of the actual scientific knowledge that has been incorporated. And that would be a start. It's a simple start.
Just a quick follow-up on this. So what kind of worries me about this is I'm thinking of the Psych project that Doug Lennart had, in which the goal was to sort of have a way to represent common knowledge about the world. And I guess it's still going on, and it's been going on for like 15 or 20 years. And so a claim seems something that's very general. And I'm wondering if solving that might require like a way to represent common knowledge, which is, you know, very intractable, or do you think that it's something that's more well limited and it could actually be achieved?
I think it can be achieved. And this is where RRIDs, identifiers, kind of come into play, right? If you think of Wikidata, it represents entities of all kinds of things, right? And if a simple thing was we provide identifiers to thing, as Anita said, the details are behind that identifier, but the identifier captures that one thing. Now knowledge becomes a set of statements about those identifiers. And that, I think, is a tractable place to start in representing knowledge.
So there's been some chatter about publications, journals, that kind of thing. They obviously could play a role in standardizing. You know, we're moving towards more open access and even other terms that my colleague JB, I think he's the one that coined data publishing, but I could be wrong. Could have been Marianne or who knows. But you know, it's not just about publishing your article, it's about publishing the data, and they could drive a lot of this. I'm aware and part of a couple of initiatives to try to, you know, facilitate this entire process, at least sort of the preprint process. So what can journals do to facilitate our quest to have more standardization, maybe with funding? Anyone want to answer that? Anita, feel free.
Speaking to a lot of journals, a lot of the time, you know, we have to kind of understand that, yes, they have a fairly large stick, but they don't like to use a stick, right? They really don't want to be able to do that. They really don't want to be seen as the people with large sticks, right? The other thing that we need to recognize is we could talk about what they can do. But as we found with RRID, something so simple, it still takes the journal a lot of time to actually enforce anything like this. Sending people to just the instructions to authors is something relatively easy and trivial theoretically, but that still takes many hours of work from them. And if there is, you know, the higher enforcement journals, they actually devote significant resources to this. So in preprints, we have a set of automated screening tools that is actually running on top of all of the COVID preprints, which has been a lot of fun. We get a lot of complaints, well, not a lot of complaints. By percentage, it's a tiny, tiny fraction, but we do get some complaints about how badly our tools had performed on a particular paper, which, you know, which leads to some ire. But those are the kinds of things that could potentially lead to increases in the ability to standardize some set of literature, but none of those things come without cost, right? So either the cost is borne by the journals, it's borne by the authors. So if there's an automated means, then they're borne by the authors, because the authors are often seen as being graded unfairly by a tool that, you know, obviously has false positives, false negatives. So I think there is a cost to all of these standards. It's just who is going to actually bear that cost.
I don't mind if it's the journals, but David, perhaps you have something to add.
Oh, I was going to say that I think that the funding agencies should have a line item that's above the direct costs for, you know, limits on the grant, that and maybe it has a sliding scale a little bit depending on how large a data set you're collecting, and that just gets added to every budget. And I don't know what the cost is, but say it's 100K and here's what you got to do. You know, you have to put your data into the standard format for your community, if you know, if one exists and so forth. That, you know, it should come with some rules, but it's just, you know, it's added on.
It's a very good point. I mean, funding agencies in the last few years have started to do that. I think they're still not realizing the full amount of cost that's associated with sustainability or any of these issues in standardization that we're talking about. But it's nice to see that now there are often line items of what's your plan to release your data. Liuba.
Well, just a side comment to this. So I fully agree. So on the funder side, I think there needs to be much more effort done. And actually the point of just asking for a standard, like using a standard, doesn't make it, right? It's about the money that you need in addition in order to hire someone to convert your stuff to the standard, or to actually get the expertise in your team to get this. But from a lot of funders, still it's they ask for it and then they neglect the money and actually say, well, find the funding somewhere else. So do it, but do it from your means that you have. And on the other side, it's often also, I think there is also still this misconception that it's easy to do, right? So that every student that comes along can actually take care of that, and alongside to the PhD or alongside the usual work that they have. And I think this is also still a bit misleading and underestimating actually the work that needs to be done in order to get standards implemented in a lab on a robust level. So yeah.
Lydia?
Tom's Zoom hand isn't working. I'm deferring to Tom. I yield my time to Tom.
Yeah, that's what I was gonna do.
Yeah. So I think one thing that a lot of the threads of conversation here are dancing around, especially the ones about cost, there's a cost of premature standardization as well. And sometimes, like, we're in the midst of like 3 different groups all working on bids to try and come up with how to deal with subjects and samples. And there's a lot of good ideas, but the question is, should we even be standardizing right now when we need people to work through examples, have use cases that are there? And if you try to standardize too early, you push stuff on the community before they're ready. So there's the double cost, which is one, that now you're trying to get this implemented in labs or implemented in sort of existing software. And then you also have the cost of, oops, we made a mistake, and now we're going to live with that cost for the rest of the time of using this tool. Because it, like, even though BIDS is flexible, you know, once something is in there, it's just like DICOM, it's not going to come out again.
That's right. Yeah, I mean, I guess it's a tricky thing, the line between the carrot and the stick. So definitely I see roles where you can push people, but it has to sort of make sense to their lab environment. We mentioned that. I mean, I can think of one example recently where, I won't name the journal or anything like that, but they are requiring that people make data open, which is very good, but then they're requiring that they make them open in the couple of repositories they only like. So, you know, it's kind of like, how do you deal with that? Anyway, it is this fine line between the carrot and the stick. Any other comments about the publication, like what publishers can do or what journals can do to work with us more and standardize? Luba, go ahead.
I think from the publisher side, I really like to have more concrete recommendations, right? I mean, there are different, I mean, we try to do this in eBrains as well, like trying to inform the community about standards and try to emphasize and recommend certain standards maybe more than others. But I think on the journal side, this could be done more explicitly. It's like not only publish your data on Figshare, but see that your data are actually annotated correspondingly so that some people can actually make use of them. Stuff like this, I think, is missing in a lot of journals. Others are very neat, right? Especially data journals are typically having a lot of recommendations and requirements attached with their publications. But I think that would be a good way of educating at least the community in general. Because I mean, I guess even in that small round we are sitting here, there are very many different standards that some people may know of us, some others don't. The others are more aware of their specific community standards, but they are connected actually to another one. So we recently had this about the BITS extensions for different extensions of specifications that are actually in line with a lot of things, even though they touch only at certain points. But this, I think, would be nice, like having more recommendations or central points of looking up standards in general.
Yeah, I think there are some initiatives that are already trying to do that, but that's a very good point. So I guess we're getting close to the top of the hour. Maybe what I'll do is I will ask a final question and I might start with my fellow Czech brother, if he's still on the call, Roman, since I haven't heard from him. But the question would be, what's the next thing that we need to focus on, or what's the biggest problem that we have in standardization that maybe hasn't been mentioned so far? Is there anything that comes to mind? So Roman, if you, didn't mean to put you on the spot, but feel free to respond, and if not, we'll go from there.
Yeah, just one idea that just came to my mind when we were talking about education. What I think we could do is to get some training about standards, tools, and into universities to young people, because everybody is in some kind of bubble, or it's stuck in his or her usual behavior in using some tools, making analysis, and so on. And I think that the youngest generation, they are more open to do the things in different way. So I would see some potential to really get this training to more to universities and to younger generation.
Totally good point. I agree with you. Since there's no panel after this, maybe we'll take a few more minutes just to finish off this point, but I'd like to hear from others that haven't spoken so much. Training, absolutely. What else is there that we need to do? Lydia?
Jill's Zoom hand doesn't work either.
Okay, yeah, sorry. Jill, go ahead then.
Thanks, Lydia. No, I think I was just gonna say the other thing is ultimately, again, these standards are for creating data sharing, but the data sharing is for the purpose of data reuse, right? So I think journals could ultimately also encourage publications that are higher visibility for data reuse instead of always focusing more on novel ideas. So that could be a way that kind of just indirectly, right, helps support the development of standards.
Yeah, absolutely.
And tool development.
Great point. What else? What else is there that people think absolutely need to be focused on right now? Satra.
So I'm going to take what Jill said and try to make it a little more general. I think standards are about resource sharing more broadly, not just data. It could be protocols and other things. And I think we have to keep that in mind. I think given our crowd, we often almost jump into data formats and data structures very rapidly. But we need standards for all kinds of things, including experimental protocols, and other resources, whether they're viral vectors or other things. And so we have to think about standards from a more global sense than just data formats.
Totally agree. Wow. Good points, everybody. What else is there? There's probably a few other things we need to focus on. Ben, go ahead.
Well, I know at least for NWB, what we're really focusing on over the next few years is ecosystem development. So we need tools to convert data to NWB, to analyze data, to visualize it, to publish it. You know, the standard is really only as good as the tools that allow you to leverage it. And right now, NWB has a lot of great tools under development, and they're coming online, but that needs to continue over the next few years in order for this to really reach its full potential. And I think that's probably true of other standards as well.
Excellent, thank you. Lydia, I believe you're up next, and then Jeff.
And I think the precursor to any standard is when you got a concept, give it an identifier, so that, and then there are lots of resources to put that identifier and explain it. Use protocol.io, use MGI, lots of places for identify, and that's the start. That's the precursor to standards.
Okay, cool. Tom?
Yeah, so I just want to echo what
go ahead, Tom. Then Jeff.
Sorry. I just want to echo what both Lydia and Satra have said, and it's sort of an answer to a point that Marianne made. She said, we can't ontologize the world, but we do have to get people in the habit of being as precise with the names of their the things they're trying to do research on, and the methods and techniques that they're using to do it. It's going to be a long haul to do that, but I think that that's sort of if we can do that, then we can at least try to ontologize the things we're measuring.
Jeff?
Yeah, this is kind of more speculative. I'm not sure if it could actually work, but I was thinking that if there was some way to have a formal way to validate papers. So right now, data sets can be validated to see if to confirm whether they adhere to a standard. And if there's some way to validate a paper, and what I mean by that is that, say that this idea that Satra had about representing the knowledge or standardizing the knowledge or claim in a paper, and then the RRDs in a paper are also included. So if you had some sort of tool that could be run on a paper that would then look up the RRDs, check the claims to see if they're referring to specific things, any data that's there, check to see if it's in a repository. And the end result of running the tool would say, okay, your paper is missing certain things that need to be included. So I don't know if this could be achieved, but if something like that could be aimed for, then that would be a way to incentivize people to really adhere to standards.
The answer is yes. We're working on it. But like, I'll give you some examples. Obviously, that I'm being a little bit facetious there, but I think there are tools, like journals sometimes do have these, like, proof editor type tools that do some of those things, like checking your references, that. But we're also working on, I'll lead you to a topic called NeuroLibre. So we're actually using notebooks to be able to make it easier to rerun analyses using the same environment that you had before, or knowing exactly what the environment variables are in a way that's much easier. So we're not only have a whole potential preprint review system revolving around that, we also have the infrastructure that is going to allow you to move notebooks through using Binder Hub and all the complicated infrastructure aspects, whether it's Kubernetes or Open Nebula, all these services that allow you to take notebooks, and then therefore all of your scripts and linking to the data that you have, and so others can take your notebook, rerun it on the data that you had analyzed, and they could get the same result much easier, right? So that's something that is an ongoing initiative, just one. I know there's other people doing similar things. So that's I think the community is moving there. So that's kind of nice, but we're not 100% there yet. I believe next up was Padraig. I don't know if I said your name right.
Oh, no, sure, that's good. Porrick. I just like to build on what Jeff said. One of the things I was thinking earlier was that we're never going to normal science as it has been going up until now is going to continue. We're never going to break the kind of thing that people are looking for the publication. They can publish without necessarily releasing their data. It's that PDF that they want to get out there and citations and so on. But the space that this standards can really move forward is if the things that happen to that publication afterwards are enabled because of the standards. So if you put your data in a standard, it's easier to use, great, that's fine. If you have unique identifiers, that's great, that's fine. One way to think about it would be to say, what's the most useful thing that can happen to this PDF or publication or piece of knowledge afterwards? What is needed for somebody to, in a very constructive, most useful way, to move it forward? And if the standards we're developing can do that, can move that forward, then that's the job done, and build on existing things.
Agreed. Yeah, that's the work that we have ahead of us. So maybe to close off the session, I will quickly ask the three people who have spoken already to give their summations. You can either answer this question, continue on this thread, or bring up whatever you want. So maybe Anita, since your hand's up, why don't you go ahead?
Thanks. I have a shameless plug for the automated screening working group. So thank you, Jeff, for bringing this up. I think this is a place where a lot of people are thinking about what to do with manuscripts and how to standardize different aspects of it. And I don't think any of us have all of the answers for how to do that. But there is a working group currently that I co-lead called the Automated Screening Working Group that is actually trying to get a lot of these tools together in order to screen manuscripts. And so I'm going to just shamelessly plug SciCrunch.org/ASWG. I made that the wrong thing, but anyway, it's the Automated Screening Working Group is a place where a lot of people who have screening tools can come and basically see if they would run in a single pipeline to screen a whole bunch of manuscripts. We're handling all of the manuscripts that are coming through in COVID, as I've mentioned. But this is a place where I think a lot of the community members here would really like to come together and maybe perhaps see if if the kinds of things that we could be screening for, together, right? Because again, coming together, an individual screening tool will not necessarily be able to give you information about each paper, but the kinds of screening tools, if you put them all together, it actually becomes really, really powerful set of checks on a manuscript. And so I will I'll change what that what that URL says. It's
been put into the chat, correct URL, so we're all good. Thank you for that. Thank you. Guiomar, do you have some closing thoughts about where we're going or anything else?
I wanted to highlight the point that Satra made that made me reflect, and is the fact when a standard will come invisible. So hopefully someday all this standard conversation that we are having now, it's like a given for new researchers. So this is something I like and to look forward.
And finally, I will just say that there is a standards and best practices committee with INCF where we actually go through a very elaborate process. I think Satra brought it up at the beginning, where we vet through, if you have a standard or even a best practice and you think it could be useful for the community, then we definitely go through that. And so it might be useful to get that knowledge out to everybody else. I'd like to close off with Satra saying a few words, because I want to feel warm and fuzzy and the way I do after I listen to Satra talk. So Satra, a few closing words, and then we'll go from there.
So I'll simply say there's a lot of excitement in the neuroscience community right now with all kinds of new directions in looking at fundamental neuroscience, looking at applications. And so there's a lot of space for the community to come together. And I think part of the goal of this panel and this conversation is how can people come together where, as a result of coming together, their efficiency and effectiveness for research improves. And if we can kind of say that it's neither sticks or carrots from an external part, it's kind of sticks internally. If I can do things better, it's likely to help everybody come together. So I would suggest that the reason we do standardization is not necessarily just for sharing output with the external world, but also to improve practices in our labs. And if you can just look at it through that lens, perhaps there'll be a broader group that comes together for standardization.
See, I do feel warm and fuzzy. That sounds really great. So thank you all for being part of this panel. Thank you to everyone who has stuck around to the end to watch this. Thank you to INCF for hosting this awesome conference. There's so much valuable information here that actually can be used for training and knowledge transfer in the future. So with that, thank you all, and I'll see you all out there somewhere.